My name is Catherine Hansen. I'm proudly associated with the MiNDAUS Partnership in my capacity as Research Manager for the Thomas MND Research Group. This interview is part of a series of interviews that we'll be undertaking with our MiNDAUS partners so that you get to know who they are and how they think their work will be contributed to and will enhance the work of the MiNDAUS Partnership. With me today is Professor Naomi Wray, who's a Professor of Complex Genomics at the University of Queensland. Naomi is one of our senior and founding partners, and her work is crucial to expanding MND care and research across Australia. Welcome, Professor Wray.
Hi, Catherine.
Professor Wray, as a specialist in genomics, how did you first become involved with motor neuron disease?
Well, I think I became involved when I really first joined the University of Queensland. My research has focused on genetics of many diseases and disorders. I suppose I'm particularly known for my work on psychiatric disorders. And when I joined UQ, Professor Perry Bartlett, who was the director of the Queensland Brain Institute, asked me to specifically establish a program in motor neuron disease research. So I've been working on motor neuron disease for really about the last 5 or 6 years.
Is genomics the same as genetics?
Yeah, that's a very good question, and it's very confusing to people.
Yes.
There is a bit of a difference. Let me try and explain it. So, genetics is the study of the DNA code. So DNA is in every single cell of our bodies, and it's what we call the blueprint of life. We know that DNA is passed from parent to child, so a parent passes 50% of their DNA to their child. And so while genetics is the study of that DNA code, genomics is the study of things that are associated with the DNA. So sometimes we know that there are changes, chemical changes to the DNA. We call those epigenetic. Epi means on top of. And sometimes those epigenetic changes are biological, because we know that while the DNA code is the same in every single cell, different cells work differently. So a liver cell is different to your neuronal cells. And part of that is controlled by those epigenetic modifications. But some of those epigenetic modifications can also be environmentally driven. So for example, we know that smoking is bad for our health in many ways, but actually how does it work biologically? How does it go from smoking to impacting health? Well, it goes, probably part of the mechanism is going to be from smoking affecting the DNA and making epigenetic changes. And then genomics is also about the studies of the genes expressed. So the DNA is coding for genes. Every cell expresses different genes in different combinations. And so we know that, for example, the genes expressed in the motor neurons of those affected with motor neuron disease is going to be different to the gene expression in people without motor neuron disease. And so studying genetics helps us, studying the DNA code bit of it helps us really get at causality of disease. And studying genomics might tell us about causality or consequences. And it's really studying both of them just helps us understand better the biological causes and consequences of disease.
Is there a fundamental question that you feel driven to answer with your research?
Yes, yeah, there is. So, the fundamental question that drives my research is what leads to differences between people. You just have to look at two families and you can see that there's must be genetic differences, genetic causes which are driving differences between people. So, in fact, almost any trait you study has got genetic contributions to differences between people. Well, maybe except things like getting struck by lightning. But other than that, almost anything you'd measure has got a genetic contribution. And so in my research, I particularly focus on, why do some people get disease and some people don't? Why do some people progress faster than others? Why do some people respond to treatment for a disease and some people don't? So that's the driving question. Just a small one.
Would you tell us how genes are implicated in ALS-MND?
Yes, that's a good question and a little bit complicated. Well, to start with, we know that about 5 to 10% of people diagnosed with ALS come from families where there are many affected members, and they would know they come from those families. And of course, when there's many affected members, then that's a really strong clue for DNA involvement. And so for many of these families, the DNA cause has been found. You know, it's a single place in the DNA which is aberrant in those people. And those families have actually been really, really important for ALS research, though they've provided important biological clues which have really gone into many, many different types of research avenues. But in fact, the vast majority of people who get ALS come from families where there aren't any affected family members. It really just comes out of the blue. And for these people, there's still likely to be an underlying genetic susceptibility to ALS, which compounds with other risk factors, which we also don't really know. And together, this, in some unknown way, leads to disease. So there's definitely likely to be a genetic contribution, but it's likely to be complex, involving many, many disease variants. And for these people, there's no good looking at their family members, because we know family members are hardly ever affected. Instead, it's really the particular combination of variants that people have that's important, and it's very unlikely that their children end up with that particular combination of variants, because children get DNA from both parents. So it's really about the combination of variants. And so to understand the genetic contribution to this type of ALS, we really need to be studying people who have ALS, comparing them with people that don't. And because our DNA has got millions of letters, it means it's very hard to work out which are the DNA letters which in combination are contributing to ALS. And so because the DNA is so big, we need to have very large sample sizes of people with ALS and people without. And so we've been studying the genetics of, it's really technology of the last 10 years has allowed us to conduct these sorts of studies, and they've been conducted for many diseases and disorders, and they, across the board, they're helping us understand the genetic contribution to different diseases. And so what we're trying to do is also do that for ALS.
Since becoming involved with motor neuron disease, you've been instrumental in extending genetic testing across Australia in the SALSA project. Would you please tell us how SALSA came about and what you've been able to achieve?
Well, as I mentioned at the beginning, when I was asked to start a research program on a motor neuron disease, I realized that to do my sort of research, what I need is information on DNA on people with ALS and people who don't have ALS. Mm. And I realized that within Australia, there were some groups who were collecting such data, but in fact, probably more than 50% of people with ALS weren't being given the opportunity to contribute to this sort of research. Mm. And because we need large samples, I've been involved in many kind of data collection situations where it's involving, you know, very large studies which are complicated to set up because you're needing to be collecting data in different ways, so collecting data online. And it's also, you know, as an analyst, my talents are in statistics and analysis, realizing how important it is to have consistent data and consistently collected data. And so I talked to the groups around Australia to see if we could set up this sort of consistent data collection with biological samples. And the opportunity really came with the Ice Bucket Challenge. So I think everyone will remember the Ice Bucket Challenge, and that raised about $2 million in Australia, and about just over 1 million was for research. And that was awarded for a single study, and it was to focus on this non-familial, this common type of ALS. And so we put in a proposal which was funded, which was to start the SALSA, so the Sporadic ALS Australia Systems Genomics Consortium. And, you know, $1 million sounds like a lot of money, but this was spread over 3 years, and part of that funding provided a research nurse at 7 clinics around Australia. And what we did was set up a system for data collection consistent data collection, and also biological sample collection. So the biological samples get sent to, most get sent to our lab, but to a couple of other labs around Australia. But we're all doing it in the same way. And this was a pretty complex thing to set up, but it was definitely worth it, and it's definitely been a team effort, but it's now providing a very strong foundation stone for research going forward. So when those biological samples come in, my interest is in the genetics, and so we generate genetic data. I wouldn't call it a genetic test. It's not a test, it's genetic analysis for research. But the biological samples are there for other people to access, because there's many ways that you can use a blood sample to look for different biomarkers, etc. At some clinics we are collecting biological samples every time someone comes into the clinic, and those biological samples are also important if we want to understand progression of disease. And in some clinics we're also collecting other biologicals like urine and feces, and so my long-term goal is that that should be done in a consistent way, that we get a complete biological resource of biological samples over time, urine and feces, because this really builds a research resource which can underpin an awful lot of research. One of the great things has been talking to people with ALS and realizing how they actually appreciate the opportunity to contribute to this sort of research. I think they really get it, that in this world of big data, they realize that we need these sort of big data resources to understand this terrible disease. And it's, they recognize it's a gift for the future.
When you're talking about biological samples, do you mean blood samples?
Yes, to give a blood sample every time they come to clinic, yeah.
If, for instance, I had my blood sampled today, would that mean that that sample would be retained and so could be useful into the future as the technology for testing improves?
Yes, exactly.
Okay.
So obviously everything we do is governed by human ethics, and so that people have to consent that that's what they want to happen, and that people are able to withdraw consent if they change their mind. But yes, that's the goal. The research I do today wasn't possible 10 years ago. It's been driven by technological advances, and we don't know what the technological advances of tomorrow are going to be. And so what we hope is that by setting up this biological resource, when the technology of tomorrow comes along, we're ready and can immediately use it and see what it can tell us, without having to wait to collect samples. So that's the idea. It's a resource that we're building up which is accessible to all researchers. And so the thing about collecting biological samples many times, obviously the DNA is the same. The DNA is the same in your cells in all your body throughout your life, mostly. But the blood sample can show us which genes are expressed and how that's changing over time.
If I'm a patient in a remote or rural area, am I able to contribute my biological samples, like my blood samples, somehow and still take part in the MiNDAUS and SALSA project?
That's something that hasn't been possible until now, but it's something we've actively getting the ethics on. And so because my group, I work on many diseases and disorders, we already have this set up for other studies where it's possible we send out a kit. A person would go to their GP or path lab, get the blood sample taken, and with that kit, the person who takes the blood knows how to send it back to our lab and receive it in that way. So yes, we're hoping that, and this is part of the next phase, is to make sure everybody in Australia who has ALS has the opportunity to contribute to this sort of
research. That is a fantastic opportunity. Fight MND has now contributed significant funding to making sure that SALSA can continue. What does that mean for MND care and research across Australia?
Yes, we were really absolutely delighted that FightMND have decided to continue to fund the SALSA program. I guess this, for me, it recognises the importance of my vision and the goal of my research about genetics, but I think it's much more than that as well. It's about recognising that we're building a future-proofed biological resource for all researchers to use. And, you know, nothing happens if the samples aren't collected in the clinic, and so it's really important that the FightMND money is continuing to support the research nurses in the clinic. And of course, from FightMND's point of view, you know, their ultimate goal, one of their key goals, is about bringing clinical trials to Australia, and how that means so much to people who are living with MND today. But of course, the way clinical trials work is that there's often very strict inclusion and exclusion criteria, and often those criteria, or certainly going forward, those inclusion exclusion criteria are likely to include genetic things. And so it's important that people with ALS, motor neuron disease, when they first come to the clinic, they give a biological sample. We can process it, get some basic DNA measures, and then those are useful for identifying people with the right inclusion-exclusion criteria for those clinical trials. So hopefully it's a win-win. So people contributing hopefully will get something out of it themselves, as well as contributing to the bigger picture for research. And of course, we have to thank all the people who've collected money and donated money to FightMND, to MNDRIA, all those organisations, the MND associations, MND and me, lots of those. It's been really fantastic.
I'm glad you said that, because there has been so much wonderful grassroots support for motor neuron disease research across Australia, from the Ice Bucket Challenge, and FightMND has really disseminated so much effort and money into facilitating research in Australia. There's a lot of people to thank. How do you think that the MiNDAUS Partnership will extend the work of SALSA?
Yes, well, it was fantastic that we got funding from actually the National Health and Medical Research Council to establish the MiNDAUS Partnership. And yes, a key component of that was building on what we've done with SALSA, making this national consortium, bringing all the, well, you know, the clinicians were all working together anyway, but this is a formalised way for that, and formalising the data collection, the sample collection. But the MND-MiNDAUS Partnership is more than that. It's bringing on board the Amanda, the registry, and all the other stakeholders, all the MND associations, the charities. And obviously it makes a big difference if we present ourselves, you know, across the board of all those people with one voice, and that can be very powerful in many ways, but certainly, you know, in policy, in everything to do with, you know, trying to improve you know, what's happening with this really devastating disease and making people's lives better.
Thank you, Professor Wray. It's been really interesting talking to you today. You have a gift of explaining things and making things that are complex seem a lot more simple. Thank you.
Well, thank you for having me, and thanks to everybody who's contributed to what we're trying to do.